John could hit.
Okay. Thanks, John. Thanks, Mike. I'm delighted to introduce Dr. Henry Nguyen from our laboratory for today's first talk. Henry's a graduate of Carnegie Mellon University before earning his PhD at Yale in the Department of Molecular Biochemistry and Biophysics, where he was trained in structural biology by Yang Shang and made fundamental contributions to our understanding of HIV host pathogen interactions, among other things. And then I was delighted when he joined our lab approximately 4.5 years ago to begin working on a membrane-bound escort-3 assembly. And with that, I'll turn the time over to.
All right, thank you, Adam. Thank you, John and Mike. I'm excited to tell you about my cryo-EM reconstructions of escort-3 filaments. So one of the major focuses in the Frost lab is understanding how membranes are reshaped or remodeled. And one of the major pathways in doing this is the so-called escort complex, the escort pathway. And escort stands for endosomal sorting complexes required for transport. And these drive membrane remodeling events throughout the cell with a whole host of different cellular processes. They were first discovered by the Emmer lab, who will be giving the second talk, and for formation of multivesicular bodies. But they have been implicated and shown to be involved in cytokinetic abscission, so the final stage of separating two daughter cells, as well as being hijacked by a variety of viruses. And the proteins that I'm going to be talking to you today are involved in some membrane contact sites, notably between the ER and the endosome to drive endosomal fission, as well as lipid droplet peroxisome contact sites. So within the escort pathway, the escort-3 proteins are the main proteins that drive membrane remodeling.
Oh, Henry, I'm sorry to interrupt, but your screen's coming out really blurry for some reason, even though it wasn't before.
Let me try something real quick. Is that any better? Or is that the same?
It's still blurry. Scott Emmer just said to stop sharing and reactivate.
Okay.
Yeah.
Still blurry. Just stopped.
No.
No? Wow. Sorry about that, everyone. Okay. Future don't do the screen optimization. So I was just saying the escort-3 proteins are the ones that are thought to drive the main activities for remodeling membranes. And then in humans, there are 12 escort-3s. They're called chimp proteins 1 through 7 with different isoforms, as well as IST1. They all adopt this canonical escort-3 fold. So crystal structures have shown that these 5 alpha helices pack together into this closed globular shape. And it's thought that these proteins must transition from a closed to open state, polymerize, and remodel the membranes. And of the escort-3s today, I'll be talking to you about 2, chimp1b and IST1. So people have been looking at escort-3 polymers for quite a while. And these are a couple of EM images of escort-3, either homopolymers or heteropolymers. And you can see they adopt a wide range of shapes and sizes. You can have some linear polymers, and if you start combining them together, so they start forming filaments, as shown up here on the top screen. And these filaments can even wrap around and form what we call these macaroni structures. These are all what we've seen in vitro. We also see similar things in vivo. So here I'm showing you a storm IF image of IST1 during cytokinetic abscission. And you can see that these things form these kind of loosely packed long coils as well. So SCOR3 proteins drive what we call positive and negative membrane curvature. What I'm showing you here now is this is a deep etch EM image from Phyllis Hansen. This is a flat membrane. The canonical activity of SCOR3s is to stabilize negative curvature. So when this protein, in this case GIM4A, polymerizes, this drives membrane tubulation into the screen. This way, the protein is now binding the lumen of the newly formed vesicle, and this is what we call negative membrane curvature. And then recently, Phyllis and others have shown that there's a subset of SCOR3s that stabilize the opposite topology. In this case, when this protein, GIMP1B, polymerizes, this drives tubulation outside of the plane of the board. And this way, the protein is coating the outside of the newly formed vesicle. So the rest of my talk will be focusing on this, this topology, this positive membrane curvature. So back in 2015, it was still unknown how any SCOR3 protein forms a polymer. And then Adam, in collaboration with the Sundquist lab, was the first to solve a high-resolution near-atomic model of a SCOR3 copolymer, showing how these things polymerize. This copolymer showed us two really interesting things. First, we formed two layers. So the first layer was comprised of GIMP1B. This is a top-down view of this filament. And GIMP1B was in this open conformation. So we like to think of GIMP1B as an arm, where it starts from a shoulder, goes to the elbow, and then has a hand region at the C-terminus. This is what an open SCOR3 looks like. Then what was really surprising too was bound to the outside of this, and the second layer was IST1, and IST1 was in this closed conformation. So this showed that both open and closed SCOR3s can polymerize together and form these filaments. Now, when we're looking at this filament some more, you notice that this thing is highly basic, so to stabilize positive membrane curvature, but it's also really, really narrow. So the distance in the lumen of this filament is 12 nanometers. So if you're trying to fit a membrane in there, that would be pretty tightly squeezed. So this is where I started to join the lab, and I was interested in how do these two proteins, GIMP1B and IST1, work together to remodel membranes. To do this, I had to do what we call membrane remodeling reactions. So in my case, I took liposomes, I mixed it first with GIMP1B, the first protein, then added the second protein, IST1, and watched how these things constrict and squeeze liposomes, our model membrane. I had to do a lot of optimization of this reaction. The key thing to do was to vary the lipid composition. It turns out we needed a lot of acidic phospholipids to help the protein bond to these membranes and remodel it. So in this case, around 5% of a FIT2-like molecule was enough to get really robust reactions. So on the left here, I'm just showing you what a cryo-EM image of a liposome looks like. I started with liposomes that are about 50 plus nanometers in diameter. When you add GIMP1B by itself, that alone is enough to take these spherical liposomes and wrap them up into a cylinder. So again, this is a cryomicrograph of the GIMP1B membrane filament. Then you can add IST1, and that will constrict that filament even further. So here is also a cryomicrograph of what we call the copolymer bound to the membrane, and you can even see a little liposome peeking out at the end of this filament. From the particles from these micrographs, I was able to generate really nice 2D averages, and you can learn a lot from the 2D averages itself. For instance, you can see the protein layer here, and this is the outer leaflet, and this is the inner leaflet. But now I'm going to show you the 3D reconstructions of these two structures. So here is a 6-angstrom cryo-EM reconstruction of the GIMP1B only membrane-bound filament. On the left is a top-down view of this filament. So you have GIMP1B on the outside, and one protomer highlighted in dark green. And then here, this is the outer leaflet. This is the inner leaflet. If you rotate 90 degrees and take a view from inside the tubule out, you can see GIMP1B is in this nice open conformation. So this filament is about 28 nanometers from outer edge to outer edge, but if you're just looking at the distance between the outer leaflet head groups, that's 18 nanometers. Now remember, we're starting with liposomes that are 50 plus nanometers in diameter, and we've already shrank it about twofold and greater. And then the distance between inner leaflet head groups is 12 nanometers. Now let's take a look at our copolymer bound to the membrane. So again, when you add IST1, that binds to the outside, and that leads to a constriction of this filament. This is a top-down view. IST1 is on the outside, as we saw before. CHIP1B is on the inside in this open conformation. And then this is the outer leaflet, and this is the inner leaflet. So this filament is 24 nanometers in diameter. But owing to the fact that you have two protein layers, the distance between the outer leaflet has decreased from 18 down to 10 nanometers. Moreover, the distance between the inner leaflet head groups went down from 12 to 4 nanometers. 4 nanometers is really, really tight. That should be at the point where you're getting hemifission of the inner leaflet. But we think our protein coat is stabilizing this highly curved bilayer. What's also interesting is that I was able to get actually two different states of this highly curved filament. If you can tell, it's kind of hard to tell, but trust me, the filament I'm showing you here is a left-handed filament, so it twists as like a left-handed screw. I was also to make a reconstruction of a right-handed filament, which twists like a right-handed screw. So this was really perplexing to us. You want to understand how can this same protein adopt two different filaments. I'm talking about filament handedness, not protein handedness here too. So here's a protomer of the right-handed CHIMP1B from the filament. Here's a protomer from the left-handed filament. If you do an overlay based on the end terminus, you notice there's a slight hinge right in between the shoulder and the elbow. And so again, we like to think of CHIMP1B as an arm, and it's kind of like the rotator cuff joint of this molecule. And the way we try to think about this is when it's polymerizing on a membrane, as it's finishing one turn, it can either continue the turn upward or downward, and that's what's dictating filament handedness. This highlights that this protein is flexible. CHIP1B is flexible. This left-handed and right-handed filament has also been seen in other escort3s as well. Moreover, it's CHIP1B that's dictating this handedness and not IST1. So here I'm showing you the EM density of the IST1 layer in both the right and the left-handed, as well as two protomers. If you take these two protomers and you overlay them, the RMSD is very, very small. It's less than an angstrom. The reason IST1 doesn't discriminate between the two-handed filaments is because it's always interacting with the C terminus of IST1 past the elbow, past where that rotator cuff joint is. So here are the protomers from the right-handed and left-handed filaments. And if you do an overlay, the RMSD is again around 1 angstrom. But if you do a focused alignment just on the C-terminal end, they overlay rather nicely with RMSD less than 0.7 angstroms. All right, so how do these two proteins actually work then to constrict the membrane? So let's first take a look at CHIMP1B. So what we see in all our reconstructions is that CHIMP1B always intertakes in this I+4 conformation. Here I'm showing you the EM density from the monoconstriction state of CHIMP1B only, and I've docked in 5 protomers of CHIMP1B. What you'll notice is that the I protomer swaps past the +1, +2, +3, such that the hand of the I protomer grabs the shoulder of the I+4 protomer. We take a look at our high constriction state, we see the same thing. This I protomer swaps around the I+1, 2, 3. Again, the hand of the I protomer grabs the shoulder of the I+4 protomer. What this is telling us is that these subunits aren't disengaging from one another. They're always interlocked in the same number of subunits. They must work in concert. Now, if we take a top-down look view of these filaments of just the CHIMP1B layers, you can see CHIMP1B is able to adopt a wide range of curvatures. And to understand how it can do this, I'm going to show you a morph movie between the protomer from the monoconstriction state and to the high constriction state. And what you're going to see is that to be able to adopt these high curvatures, CHIMP1B just has to flex at the elbow to adopt this different degrees of curvature. And it's a subtle flex in the elbow that allows CHIMP1B to do this. What again this shows us is that CHIMP1B is flexible. I think the flexibility of CHIMP1B can be further extended to how we think about the other escort3 proteins. So as I've shown you before, these things can adopt a wide array of shapes and sizes by themselves. Now, the consequence of CHIMP1B flexing at the elbow and also always being interlocked in an I+4 configuration is that as you're constricting this membrane, you're also going to elongate the membrane. So here I'm going to show you a movie where I apply that simple elbowplex across an entire turn of chimp-1b molecules. And as you can see, that as it's constricting, you're going to get elongation along the filament axis. And what I also want to point out is that, as you can appreciate from this movie, that there's really little contact in between rungs, that these subunits are easily sliding between turns. And I think this is also important to help us understand what we see in, in vivo. Again, this is the STORM IF image of IST1 during cytokinetic excision, and these loosely packed loose coils that we see in IST1 also help support the idea that there are very little contacts between S-squared 3 rungs in filaments. All right, so now we understand how chimp-1b is working. So we know IST1 is driving this constriction, but how? And it turns out that IST1 is homopolymerizing upon the chimp-1b strand to cause that constriction. So here I'm showing you 5 protomers of chimp-1b from the moderate constriction state. I'm going to dock in how I think IST1 initially binds to this filament. It binds again to the C-terminal end, as I talked about earlier. So this is how one protomer of IST1 would bind. This is how a second one would bind. And now I'm going to dock in how 2 IST1 protomers are actually interacting from our high constriction states. So as you can see, there's a swinging of subunits, slight swinging, that occurs upon binding. And this slight little hinging of IST1 is what causes chimp-1b to flex its elbow, to constrict the membrane, and elongate the filament. All right. Now, let's take a look at what's actually happening at the membrane. So here's again a top-down view of our filament. And now I'm going to switch to a grayscale view to help highlight better the membrane. So this is the outer leaflet, that's the inner leaflet. This is a rotation 90 degrees and a slice along the filament axis. So this is the inner leaflet, and this is the outer leaflet. And I hope you can tell, whenever the protein is interacting with the membrane, there seems to be little divots or deformations that we see at high curvature. So I'm going to zoom in here. So again, at the outer leaflet protein membrane interface, you see this little divot. And this is caused by helix 1 of chimp-1b, that is the helix that's interacting with the membrane. So we have 3.1 overall resolution, but I just want to show you, this is the actual density and model for helix 1 at the protein membrane interface. And we have quite good density there. So you can see a lot of well-resolved side chains even at that helix at that protein membrane interface. I want to highlight, this is the face of the helix that's interacting with the membrane. We see an arginine and lysine, so very basic residues. That makes sense to complement the acidic membrane. What we were surprised to see was the fact that 2 phenylalanines are also at the membrane. And we think those may be important for stabilizing membrane fission or downstream events. Finally, one of the last things I want to talk about is that as a function of constriction, we saw that the bilayer thickness was decreasing, or the bilayer was thinning. So we measured our starting bilayer thickness to be around 37, 38 angstroms. Then we measured the bilayer thickness from our moderate constriction state by measuring the distance between the inner leaflet and the outer leaflet. And already in that first stage of constriction, we've decreased from around 37 angstroms down to 30 angstroms. Then if we look at our high constriction states, where we have 2 of because the right and left-handed filaments, we see that this trend also follows that as you squeeze it even further, the bilayer decreases from 20 down to 28.6 and 28.4 angstroms. And also as a consequence of this, that the inner leaflet becomes more dense as you're squeezing it. So some future questions that we'd like to address is, 1, how are the lipids behaving at this such high degree of curvature? But ultimately, we want to figure out what happens. How do you go from this state to a state that leads to membrane fission? But those are, future experiments for a later time. So I just want to summarize with this nice movie of what our reconstructions have shown you, is that chimp-1b alone can deform the membrane, and then as IST1 homopolymerizes on that chimp-1b layer, that drives chimp-1b to flex at the elbow, constrict the membrane. And with that, I like to go to my acknowledgements. Thank Adam for my advisor, giving me this opportunity, as well to his two co-host collaborators, Nathaniel Telledge and John McCullough, as well as the Verschining Lab, who did some experiments that I didn't have time to show you, as well as the Grabe Lab, who are membrane biophysicists that we had a lot of discussions with, the Cheetah Center for funding, and the UCSF EM Core for microscopy help. With that, I'll take any questions.
Okay, thank you so much, Henry. The chat is open for questions. I'll be monitoring the chat window for people who want to type them in. John Burke has got the first question for us.
Henry, really cool talk. So a couple of quick questions on that switch from the right-handed helical filament to the left-handed helical filament. So first off, have you tried making any kind of mutations in that area to see if you can lock it in, or playing around with that flexibility? Or is that so much controlled by IST1 that you can't really change the inherent flexibility of the chimp?
So two things I want to clarify. There isn't really a switch between the left or the right-handed. Is that once the polymer is forming, it either decides to go left or right-handed, and we think that's pretty stochastic. I looked regarding chimp-1B flexibility, looking at those joints. There are no clear residues that really kind of explain why that it should be a joint or why it is flexible. And so we haven't been able to, I haven't made any mutations to try to lock it in one form or the other.
We have a question from Pietro asking for you to share a little bit more of your thinking about that pair of phenylalanines scooping out the membrane.
Right.
Yeah, can I comment? Can you hear me? This is Pietro. Can you hear me? Yeah, because if I remember correctly, also in the Marx protein, you have a lot of basic amino acids and several phenylalanine, and also another membrane interfaces. You see that. So what may be the general significance?
We think that it might play an important role for when you even tighten further. So at the current stage, we see the protein is only at the membrane interface. It's not inserting into the membrane. So potentially that these phenylalanines may actually insert into the membrane, maybe perhaps destabilize the membrane that will eventually lead to fission. That's a possibility.
Maybe I'll just elaborate on that just a tad. At this, I think what Henry said is exactly right. At this degree of constriction, and we don't know what happens at higher degrees of constriction. At this degree of constriction, one model would be that those phenylalanines are displacing the polar head groups and getting quite close to, say, the glycerol backbone, or perhaps even a bit lower. And the hydrophilic head groups that have been pushed or splayed apart, especially considering the curvature of that leaflet, may then be interacting with the neighboring charged residues. But this will require further work.
Yeah. So I'll say we haven't trapped an even tighter constriction state yet, but that is perhaps maybe we need an additional factor for that to occur.
Thank you.
Okay, David, go ahead.
Hi, thanks. I was just wondering if the limiting factor on the size of the constriction is due to the proteins being at their maximum constriction, or whether it's due to the clustering of the head groups of the inner leaflet.
That's a great question. I think there will be a limit to how tightly these proteins can constrict based on their spheric clashing. And I think there's also a push and pull between the protein and the membrane. And the membrane doesn't want to be so highly constricted, and it'll probably be pushing outwards. We haven't been able, as I said earlier, we haven't been able to get an even tighter constriction state. I think that eventually the filament adopts the preferred geometry of one of these proteins. I didn't get to have a chance to talk about that, but if you see our paper, we have some ideas about that. Basically, yes, there's some steric hindrances preventing from the proteins, preventing even tighter constriction.
And it's probably worth pointing out that this radius of this degree of the size of the lumen is very similar in our empty structure as it is in this structure. Okay, we have a bunch of other, so let's see. Thank you, David. Anirban, that's a cool parallel.
Yeah, sure. I mean, that's not a question. Just wanted to sort of just mention that. Yeah, I think we should move on to the next question for the sake of time.
Okay. John, please go ahead.
Great talk. I was just wondering if you could maybe elaborate more on the sort of composition of liposomes you use and sort of that optimization. You said the acidic composition was really important. See that assembly?
Right. So I end up using a lot of different lipid compositions, and at the end of the day, having at least 5% of a PIP2 was important. I also, in this mixture, included a lot of polyunsaturated lipids, with the idea that Bruno Anthony has some papers saying that that would help deform the membrane. But the bigger factor was this PIP2, and the way we rationalize that is that because the lumen of CHIMP1B is so basic, you need to increase binding of CHIMP1B to the membrane. You want even more acidic head groups there. For instance, if you have 100% just POPS liposomes, you don't get really robust activity. Like, you need to boost up the charge with these PIP2 molecules. And it's pretty agnostic to the type of PIPs. So you can use PI4/5, PI3/5. I don't think, at least from this, that there's any selectivity there. I think it's purely just based on charge.
Sudeep has a question about timing.
Hi, Henry. That was a great talk. Thank you. I was wondering if your model implies that there's a kinetic lag between the recruitment of CHIMP1 and IST1 on the membrane. And if so, would it be possible to test this in vitro or in cells, or would we not have enough time resolution or temporal resolution to test that idea?
Yeah, I think it's a great question. I don't think we have the temporal resolution. Just for instance, in how this reaction is done, the initial step of CHIMP1B remodeling is quite slow. That takes overnight. But once you form the CHIMP1B membrane-bound tubule, the binding to IST1 and the constriction happens in minutes. It's in the order of like 5 minutes, and already you're going to get robust tubules.
Thank you.
Okay, Kale?
Hi, very nice talk. I was curious about the ratio of lipid molecules numbers in the inner and outer leaflet and what happens during constriction.
That is a great question. So we're beginning to address those questions with some MD simulations with the Grabe lab. And it's possible. So all we can tell is that the inner leaflet gets enriched as you constrict. Whether or not lipids are flipping in and out, it's still being, we don't quite know yet, but it's something we're taking a look into. I think it's important to note that there's always a lipid reservoir from the rest of the liposomes to be a source of extra lipids as well.
Okay, and if we still have time, Guy Pearson asked me to read out his question about spastin and the C-terminus of CHIMP1B. Can you say a bit, Henry, about where spastin would engage your structure?
Yeah, so where does spastin bind? And this is dependent on the polymerization state of IST1. So where spastin would bind IST1, it would be on the outside of the filament. It basically pokes where spastin would bind out over here. And it does not depend on the polymerization state of IST1. The MIM, or the motif that binds spastin, there's a long disordered region that pokes out from IST1 into the cytosol that spacin could bind. And I think it doesn't require the polymer to form, but once you have the filament, spacin can bind, multiple spacins could bind that would have an avidity effect to help form a hexamer of spacin.
Okay, I think it's time to transition to our second speaker.
Okay, Adam. Thank you. I'm Scott Emmer, and I'm going to introduce Jeff Jorgenson, who has been a graduate student in my lab. Jeff did his undergraduate work at the University of California at Santa Cruz, and he's been in our lab for 5 years. Jeff recently, 2 months ago, defended his thesis successfully, so he's Dr. Jorgenson now. He has a great mix of traits, but two important ones are Jeff is a dreamer, he's an idea generator, and has been a lot of fun to interact with and come up with new challenges. He also has great insights into tools and approaches, and he's done 3 projects in the lab. One has involved looking at membrane protein quality control. Another has been to characterize a novel protein at a membrane contact site. And today he's going to tell you about a genetic approach he's used to try and understand how plasma membrane ER contact sites regulate lipid homeostasis. Jeff, we're ready.
Thanks. I'm assuming everybody can see the screen and that's fine. So since this is such a large and diverse audience, I thought that I would start my talk So as the Earth was cooling and the oceans were a primordial soup, the first cells emerged. And these cells were probably extremely simple. Basically, a membrane which enclosed a mixture of enzymes and genetic material. And these cells grew and divided, living a very simple life for a long time, until they started to compartmentalize their cytoplasm into specific membrane-bounded organelles. And this increase in complexity and organization has let eukaryotes evolve extremely rapidly throughout their relatively short evolutionary existence. And now, when you look around you and you see living things, basically everything you see is a eukaryote. Now, organelles are beautiful inventions of eukaryotes. You can see that each organelle has a specific biochemical microenvironment, and those are tailored for each organelle's specific function. Organelles allow eukaryotic cells to increase the efficiency of many biochemical reactions, and they do this in a couple ways. They can increase substrate concentration in the lumen of an organelle, and perhaps more importantly, they can physically separate antagonistic reactions. For example, in the cytoplasm, the cell synthesizes fatty acids. However, beta oxidation of fatty acids occurs in the mitochondria or in the peroxisome. And if we take a closer look at an organelle, there are 2 very important features. One is the membrane, and this provides the physical separation between the organelle's lumen and the cytoplasm. And in this cartoon, I've way oversimplified the organelle membrane. Here it looks like one species, but in reality, it's a complex mixture of different lipids. And each organelle has its own specific complex mixture of lipids. And that helps give each organelle its identity, and it also helps facilitate the function of each organelle. The other major component of organelles are membrane proteins, which facilitate the selective import and export of molecules between the organelle's lumen and the rest of the cell. Now, organelles are obviously advantageous to eukaryotic cells, but they're also a logistical nightmare. The cell has to be able to coordinate the function of all of these organelles, as well as deliver the correct lipid species to each organelle membrane, as well as the correct proteome to each organelle. Now, the secretory pathway is probably the best described method that the cell uses to accomplish this. And it begins at the ER, where most of the cell's lipids are made, as well as where membrane proteins and luminal proteins are translated. From the ER, proteins destined to other organelles are sorted and packaged into small vesicles that bud from the ER membrane using these coat proteins. They form vesicles which can then go on to fuse with the Golgi. Once at the Golgi, other sets of coat proteins recognize different proteins and package them into specific vesicles that go on to their terminal destinations. Now, it's important to realize that all these different coat proteins that are providing the selectivity are mainly interacting with other proteins in the membrane, and they're not necessarily sorting phospholipids. So the secretory pathway, while it's a wonderful method for delivering proteins to each organelle, it doesn't it can't account for the specificity of the lipidome of each organelle. Now, membrane contact sites have been observed for a very long time. They were first observed in the 1950s from electron microscopy experiments. But it hasn't been until the last 15 years or so that their role in intraorganellular communication or non-vesicular lipid transport has really come under light. And as you can see from this cartoon, the ER is the major organelle that makes contact with basically every other membrane throughout the cell. And this is conserved throughout all eukaryotes. In our lab, we use yeast as a model system. And in yeast, the ER makes extensive contact with the plasma membrane. Nearly 40% of the plasma membrane is in contact with the ER. And these contact sites you can see in this electron micrograph that I made. You can see a sheet of ER closely opposed to the plasma membrane. Now, these contacts aren't just random. They're facilitated by specific tethering proteins that mediate the close apposition of these two membranes without allowing for fusion between them. Now, our lab has spent a lot of time studying these ERPM contact sites, and we've characterized many of the proteins that are required for tethering the ER to the plasma membrane. We found that there are at least 6 proteins that are very important for maintaining this contact, and they're all conserved from yeast to humans. They're the extended synaptotagments, which in yeast are TCB1, 2, and 3. There's a TMEM16 protein, which in yeast is IST2, and there are the VAPs, which in yeast are encoded by SES2 and 22. Now, it's also important to note that these tethering proteins also have domains involved in other functions. For example, the TCB proteins have an SMP domain, which is thought to potentially facilitate the transport of lipids between membranes. IST2 is a putative ion transporter, and the VAPs can interact with oxysterol binding proteins, which are known to facilitate sterol trafficking throughout the cell. Now, normally a yeast cell has much of its ER in contact with the plasma membrane, but we found that if you delete all 6 of these tethering proteins, the cortical ER is mostly lost, and you gain this sort of cytoplasmic tangle of ER. And you can nicely see this using electron microscopy. So in this micrograph, the ER has been false colored. You can see the blue represents cortical ER, green is cytoplasmic, and purple is the nuclear envelope. You can just appreciate how much of the ER in yeast is in contact with the plasma membrane. However, if we delete all 6 of these tethering proteins, generating a strain that I'm going to refer to as the delta tether strain, the ER morphology changes drastically. It loses almost all of the cortical ER that's in contact with the plasma membrane, and you gain this tangle in the cytoplasm. And interestingly, you can see in this micrograph, the ER is still making contact with other organelles in the cell, for example, like this mitochondria. It's just lost its ERPM contact. So something I found really interesting about this was that you delete 6 highly conserved proteins and change the ER's morphology drastically, but the delta tether cells still grow like champions under normal laboratory conditions. On both YPD, which is a rich media, or SCD, which is a minimal synthetic media, the delta tether strain grows essentially indistinguishably from wild type cells. So I've been really interested in what the real important function of ERPM contact sites are in yeast. And I hypothesized that there must be other pathways that are compensating for the loss of these contact sites. And I decided to use a genetic approach to identify synthetic lethal genes in the delta tether pathway, delta tether strain. And infer the function of those synthetic lethal genes to understand what BRPM contact sites are doing. Now, since the delta tether strain is such a complex genetic background, it's not really a good candidate to do synthetic genetic arrays. So instead, I used what's called the SAT assay, and this stands for Saturated Transposon Analysis in Yeast. And this assay was developed by Benoit Kornman's lab. And when I first read about it in their eLife paper, I thought it was a beautiful assay that I wanted to be able to use it. So the assay is really simple and elegant. It's based on a plasmid, which contains an Ad2 ORF, which is required for making adenine. However, this ORF is disrupted by a DS transposon. The plasmid also contains a transposase, which is on an inducible promoter. So by inducing the transposase, you can remove the transposon from Ad2, and it inserts randomly into the genome. The yeast cell can then repair the Ad2 ORF, allowing them to make adenine, which allows them to grow on media without adenine. And this way we can select for cells which have removed the transposon and put it into their genome. Now, the way we use this to identify synthetic lethal genes is based on something called survivor bias. And it is a little bit counterintuitive, but there's a nice story that's made famous by a statistician named Abraham Wald. And the story goes like this. During World War II, the United States Air Force commissioned a group of statisticians to study returning planes that returned from war, and study the pattern of the bullet holes on those planes. And the goal was to identify the most efficient places to add armor to these planes. Now, they superimposed bullet holes from all these planes that returned from war, and they noticed the wings, the fuselage, they have tons of bullet holes. And at first they thought, these are the places that are most easily hit, so we should apply armor to them. However, Abraham Wald realized that there's a massive survivor bias in the data that they're using. And that is that they're only analyzing the bullet patterns on planes that made it back from war. And the bullet holes that they're really concerned with, or should be concerned with, are ones in the engines, the cockpit, places where they don't see bullet holes in returning planes. So in a way, in the SAT assay, transposon insertions are bullet holes. And instead of a plane, we're tracking where they're inserting into the genome. So we're looking for specific sites that are lacking transposon insertions in the delta tether strain, but not in the wild type strain. So I generated massive libraries of transposon mutagenized cells in both wild type and delta tether backgrounds. And I sequenced over 100,000 unique insertion sites in the delta tether background and over 240,000 in the wild type background. And here in this table, you can see each of these black lines represents a unique transposon insertion site. And these big black bars up here are ORFs in the yeast genome. And what we're looking for are genes just like this, where there are no transposon insertion sites in delta tether. However, there are in wild type, indicating that this is a synthetic lethality in the delta tether background. I found that using these scatter plots is the best way to sort of visualize this data. So each dot represents a different ORF in the yeast genome, and its position on the plot is dictated by the number of transposons sequenced, number of unique insertion sites sequenced in the wild type background or in the delta tether background. So by drawing this trend line, we can assume that ORFs that are close to this trend line are equally sick or healthy when disrupted in the wild type or delta tether backgrounds. Now, since we're really interested in ORFs that are synthetically lethal or sick to delta tether cells, we focused in on this region here. Now, I tested many of the best looking hits, but I found that they were false positives. And this was because the delta tether library was relatively small, so it was pretty noisy. So to increase my chances of finding true positive hits, I searched for groups of genes that function in similar pathways. And when I did this, I found a group of VPS genes, and VPS stands for vacuolar protein sorting. But this group of VPS genes also, they're all components of the escort complexes. This is something we just heard a lot about. In yeast, the escorts are primarily responsible for the degradation of membrane proteins, where on endosomes, they sort ubiquitinated cargo and then facilitate the internalization of these cargo into small vesicles that bud into the lumen of endosomes, generating multivesicular bodies. Eventually, when these multivesicular bodies fuse with the lysosome or vacuole, they deliver their internal contents for degradation. And the escorts are actually a set of protein complexes where the early protein complexes, escort 0 and 1, bind and sort ubiquitinated cargo. Escort 2 helps nucleate the escort 3 filament, which helps corral the cargo and provides the deformation and membrane scission force to generate these interluminal vesicles. And importantly, all of these escort components are required for the multivesicular body pathway to function. So next, I tested whether or not escort mutants gave a synthetic lethality in the wild type or delta tether background. And when I did this, I was really disappointed because I found that none of them gave any growth defect at all on rich media or YPD. However, the Satay assay is done on a synthetic media, so I decided to test whether or not they're sick on synthetic media. And when I did that, I found something that was really surprising, and that was that only SNF7 mutants were synthetically sick in the delta tether strain background. And this is really weird because there's no known pathway where only SNF7 or only escort 3 components are required for function, indicating that this may be functioning through some sort of new, not yet defined pathway. Now, because it was so strange that only one of these escort complexes was synthetic lethal, I thought I must have messed something up. I made this mutant over 10 times and complemented it. It fits. So I could, sleep easy at night. So to try to understand what's causing this synthetic lethality, I thought that if I could identify the nutrients in rich media that suppress the growth defect that's seen in synthetic media, that could help me understand what's going on in these cells. Now, I decided to first start by looking for nutrients that are in the lipid synthesis pathways. And this is because the ER is the site of lipid synthesis, and ERPM context sites have been implicated in lots of lipid transport or sensing activities. So these are growth assays of delta tether and SNF7 mutants, and you can see how well they grow compared to delta tether SNF7 synthetic sick mutants. Now, I found that if I supplement in choline or ethanolamine, it gives a very strong suppression of the growth defect of the delta tether SNF7 background. However, when I supplement in serine, it doesn't affect growth at all. Now, because choline and ethanolamine feed into the Kennedy pathway to make PC and PE respectively, I decided to look at the lipid pools in these cells. I hypothesized that maybe they were deficient in either PC or PE, or they were accumulating some sort of toxic lipid intermediate. So I extracted lipids from cells and I ran them on thin layer chromatography plates to separate and visualize them. And as you can see, wild type and delta tether cells have fairly similar lipid profiles. Delta tether certainly has lower PS levels and higher triacylglyceride levels. And when I looked at the SNF7 mutants, their lipid profile looked very similar to wild type cells. However, when I looked at the delta tether SNF7 synthetic sick mutants grown in synthetic media, their lipid profile was very strange. They had very low levels of PS, very low levels of PE. They accumulated large amounts of free fatty acid, oleic acid primarily, and they also accumulated tons of triacylglyceride. And importantly, when I complement this strain with SNF7, the lipid profile goes back to that of a delta tether. Now, because the triacylglyceride levels were so extreme in these cells, I thought it would be interesting to visualize the lipid droplets in these cells. So I used BODIPI, which stains neutral lipids. And as you can see in wild type and SNF7 mutants, they have roughly similar numbers of lipid droplets per cell. And these cells are grown in mid-logarithmic phase. However, the delta tether cells do have more and what appear to be brighter lipid droplets, which is consistent with what I saw in the TLC plates where I saw more tag. And then when I imaged the delta tether SNF7 mutants, they were just full of extremely bright lipid droplets. And when I quantified this using flow cytometry, I could see that delta tether SNF7 mutants had roughly 4 times the amount of BODIPI fluorescence as wild type cells. And if I supplement choline to their media, it reduces the lipid droplet fluorescence or BODIPI fluorescence to that of about delta tether cells. pretty much completely rescuing the synthetic effect. Now, because the lipid droplets look so interesting under fluorescence microscopy, I had to look at them under electron microscopy. And what I saw was that the delta tether SNP7 mutants indeed did have larger and more lipid droplets than delta tether alone. But what was really fascinating was that many of the cells had these concatenated lipid droplets where there's a neck between sequential lipid droplets. And in this single cell, you can see 4 in a chain. And this is in a 70 nanometer section. I wouldn't be surprised if all of these lipid droplets were concatenated in some way. And of course, if I supplement in choline to these cells, the lipid droplet phenotype is totally suppressed and it looks like a delta tether cell. Now, this really strange lipid droplet phenotype made me think that these cells must be producing neutrolipids at such an extreme rate that the normal machinery or mechanisms to process the lipid droplets and make them look normal was overwhelmed. So I thought that if I could maybe rebalance lipid synthesis, I wanted to see if that rescued the growth defect of these mutants. So in yeast, the balance of lipid synthesis between phospholipids and neutrolipids is achieved by what I like to call the George-Kármán pathway. And this is regulated by the availability of phosphatidic acid and DAG. And the availability of these two lipids is controlled by the activity of the enzyme PA phosphatase, which is encoded by PA1. This dephosphorylates PA1, dephosphorylates PA, making DAG, and DGK1, which is the diacylglycerol kinase, which phosphorylates DAG to make PA. Now, in the delta tether SNP7 background, it seemed like PA1 was potentially hyperactive or DGK1 was potentially underactive. But in any sense, there was too much tag being made and not enough phospholipids. So I wanted to see if I could upregulate DGK1 to make more PA and see if that rescued the growth defect. And when I upregulate DGK1 expression, I found that indeed it does rescue the growth defect, which supports the idea that in the delta tether SNP7 strain background, their lipid synthesis is messed up and they're making neutrolipids at the expense possibly of phospholipids. Then finally, I wanted to test whether or not artificial tethers could rescue the growth defect seen in the delta tether SNP7 strain background. So I found that expressing these artificial tethers, which are ER membrane proteins that have pH domains that interact with PIPs on the plasma membrane, does in fact rescue the ER morphology in the delta tether SNP7 strain. But I wanted to test whether or not it also rescued the growth defect. And what I found was that it had little to no effect on the growth of the delta tether SNP7 strain. And this indicates that of these tethers, we always call them tethers, but they have more function than just tethering. It indicates to me that the potential lipid transport activity of these proteins is what's important for the synthetic lethality seen when SNP7 is deleted in this strain. So this brings us to the model. So I think that ERPM contact sites are able to regulate lipid synthesis by transporting lipids between the ER and the plasma membrane. And I found that for some reason that is still, I can't explain, that SNP7 or an escort3 protein is required for maintaining normal lipid synthesis in the absence of ERPM contact sites. But I think that when you lose ERPM contact sites, you're accumulating phospholipids that should be transported from the ER to the plasma membrane. And the cell is able to sense this, and the ER thinks that it has too many phospholipids, so it starts making neutrolipids. And then in this background, when you lose escort3 or SNP7, this is further dysregulated because you lose this extra check, and cells start making so much neutrolipid that they're not able to make enough phospholipids. So in summary, I found that escort3 has a synthetic lethality in the delta tether strain background, and that ERPM contact sites are required to maintain proper lipid synthesis. And I think that this is due to the lipid transport activity of the tethering proteins. And I also found that escort3 or SNP7 specifically may affect lipid synthesis through some unknown, not yet defined pathway. And with that, I'd like to thank Scott members of the Baskin lab, especially Reka, who helped a lot with the lipid analysis, and the Kornman lab for supplying me with the SATE reagents.
Okay, thanks, Jeff. We'll go to the questions, and I see Adam Frost has one. Adam?
In the light microscopy, beautiful talk, Jeff, really fun. In the light microscopy images of your lipid droplets, it seemed that the single SNF7 null looked more tubular. Could we look at that again and get your thoughts about it?
I have to say that's not something that I had noticed before.
It's subtle.
There's some hazy, it's hard to tell if that's background or tubular droplets, but yeah, that's something interesting that I hadn't really considered.
George, number one, George Carmen, I assume.
Hi, yeah, this is George Carmen. Jeff, that was a really nice talk, and thanks for the plug on the PA-DG connection. I was wondering, did you ever try to see if the PAH1 mutation would rescue the mutant?
I did not use any PA1 mutants.
I would try that, because
Okay.
I bet you it would work.
Yeah, no, I mean, I think that it probably would also work. It was just a lot easier for me to just clone DGK1 into an overexpression construct to do the synthesis. But yeah, no, I think that would be really interesting.
Great job.
Tighten it up too, I think.
Roger Williams.
So it was a very, very nice talk. I was just wondering, do you know yet what's causing this constriction between the lipid droplets that are coupled together?
No, I mean, that is like one of the most interesting things to me, that actually happens. I don't understand, but it seems like there must be something around that neck. I don't understand.
You don't see anything in the electron microscopy to suggest it's protein code or something?
I don't see any density, but you know, no, I don't.
Okay.
Thanks, Roger. Dan Rapin.
Hi, one of my questions is going to come up later, but it was interesting to me that choline and ethanolamine were able to rescue, but not serine. And I'm wondering if these cells lack a phosphatidylserine decarboxylase. Does phosphatidylserine does it increase and get sort of overly expressed, or is there a large increase in PS synthesis without it being converted to PE in these cells?
I don't think so, because the PS levels are extremely low in these cells.
So it's not made at all?
No. Interestingly, when I look at the lipid profile, PC does not appear to be low in the lipid pools of these cells. However, adding phosphatidylcholine is sort of the best rescue that I've found. But I think sort of the key is that when you activate the Kennedy pathway by adding choline or ethanolamine, what it's really doing is it's sucking DAG. So it's not allowing the cell to use DAG to make neutralipids, but instead it's putting it into phospholipid synthesis and letting the cell sort of catch up with its own problem, where it's for some reason it thinks it needs to make neutralipids when it shouldn't.
Thanks, Dan. Hello, Binks. Binks Wattenberg.
Hi, Scott. Yes. Hi. Yeah. So I really like the idea of there being an ER sensor for the lipids. So I'm wondering if there is like a single protein or complex, whether a mutation of that would rescue your phenotype, and whether you had any evidence for that.
So I don't have good evidence for it. What I have noticed is that I never popped any suppressors. So, I mean, that's a little bit of evidence saying that there probably isn't a single thing that you can mutate to.
Reach XW.
Hi, nice talk. I just want to clarify, can you, is SNF7 the only component or any of SCOR3 component can rescue?
So I tested several escort3 components for synthetic lethality or synthetic growth defects in the delta tether background, and SNF7 was by far the strongest. Some of the others had mild effects, but they were quite mild, but more than any of the other escort complexes. So right now, I'm not sure if it is a very SNF7 specific function, or if it's just because SNF7 is the most highly expressed escort 3 in yeast that gives it the strongest synthetic growth defect.
All right, thank you. Nice talk.
Thanks.
Hey, Julie Brill.
Hi, so I really liked your talk. That was really fascinating. I'm wondering, I guess I can't really envision what role you think SNF7 or escort3 might be playing in regulating this process, and I guess I wanted you to speculate on that a little bit so we can try to envision it.
Yeah, I know. It's really hard for me to come up with like a mechanism that makes sense to me. It's tough. I've come up with really weird models, like in my thesis discussion, where the cell may use so SNF7 can be in a cytoplasmic or a membrane-bound form, and it seems to bind those acidic lipids more. So it's possible that the cell can sense cytoplasmic levels of SNF7, and it uses that as a sensor for phospholipid levels. But I mean, really, I haven't been able to come up with anything that makes a lot of sense. I originally liked the idea that SNF7 was involved in lipid droplet scission or somehow remodeling lipid droplets, but I haven't been able to visualize SNF7 on lipid droplets ever. And I spent a lot of time trying to see that. So,
Jay Nichols.
Hi, thanks a lot. Great talk. Have you ever looked at reducing tag levels by knocking out some genes in tag and see if you suppress the phenotype?
So I started to try that, but I found that if I deleted one tag synthesis gene, it did nothing. You have to delete a lot of them. And I've already got 6 genes deleted in this background, so it's a huge pain to make those genetic mutations. But yeah, that's something I wanted to test. And if I had all the time in the world, I probably would have.
All right, thanks.
We may have time for a couple more questions. Adam, a quick one? Oh, no, that's private. Sorry. How about George Carmen? Oh, and he's answering, I think, something for Dan. It's a connection to pi dependent. He's answering that. How about Wu?
Hi, I'm wondering where does SNF7 localize in the delta tether cells?
So its localization is mostly similar to wild type cells. It primarily localizes to endosomes. And there's also a small cytoplasmic pool. But in the delta tether strain, there's also slightly more SNF7 at the plasma membrane. And this is because in the delta tether strain, there's a pathway called the RIM pathway, which recruits escort3 to the plasma membrane. And this pathway is constitutively activated in the delta tether strain background. And it's thought to be activated because the lipid composition of the plasma membrane isn't quite right in the delta tether background. And there's a sensor that activates the RIM pathway. And for a long time, I thought that this synthetic lethality was through the RIM pathway because it made a lot of sense. If it's constitutively activated in the delta tether background, it must be required. But when I delete the RIM pathway, it has no effect. It's specifically an escort2.
Maybe just one more, James Holtzman.
Hi, that's a really nice talk. And this image we're looking at right now of these droplet contacts is really striking. And in some cell types, if you lack sufficient phospholipids like PC, droplets will undergo spontaneous fusion due to lack of sufficient surfactant on the surface. Are we looking at like an intermediate infusion? If you look at later time points, do these actually fuse, or is it actually stalled and stable at this kind of dumbbell looking?
I have no idea. I would assume that it's stable. This is just from a mid-log culture. But I don't know. I mean, I see it in so many cells that it makes me think it's not an intermediate. To me, in my, this is totally made up in my mind. I don't know the thermodynamics of it, but I would imagine once they fuse, that would happen pretty quickly.
Right. Very interesting. Thank you.
Thanks.
Okay, thanks, Jeff. Thanks, Henry. And maybe I'll leave it to Mike or John, a final comment.
Okay, well,
thanks.
Sorry, Mike.
Oh, you
go for it, Mike.
I was just going to thank the speakers. Those were excellent talks, Jeff and Henry, and everyone for tuning in. We're going to post these videos on ASBMB, and that's it. Do you have anything, John?
Nope, that's it. So just a round of virtual applause for all the speakers. That was great. And we'll see you all next week.